Hello, everybody. I think we've got everybody in here. Is there anyone still out there? Anyone want to go out and tackle any stragglers? Okay. Welcome. Thank you for being here. Thank you for coming to ASCO event, Science versus Cancer. We've got a great program for you tonight, and thrilled to have all of you in the room. The doors are shut now. I want to thank Foundation Medicine for sponsoring this event. Couldn't do it without them. And I want to thank our speakers, Rick Pazdur and Susan Galbraith, for being here. I'm supposed to make a whole bunch of pitches here right now about STAT. So does anyone not subscribe to our Cancer ASCO in 30? Raise your hand. Just Rick. Thanks, Rick. Does anyone, so subscribe to that. Does anybody subscribe to the readout? If you don't, it's fun. Does anyone subscribe to Daily Recap? It's also a good one. What about, how many people are fans of the Read Out Loud? So, I mean, if you aren't, you should probably do that. And how many of you are interested in our other live events or summits? So just to put a little bit of a plug in, we will have an all virtual summit in September called the Future Summit. The speakers will include George Church and Brian Wallach and a whole bunch of the ALS advocate and a whole bunch of other interesting people. And we are already lining up speakers for our Boston Summit in October. So if you want to know more, ask any of us. And with that, we've got a full room, which actually the whole point of me standing up here is to fill in the room. So I just want to give a warm welcome to Dr. Brian Alexander, the Chief Executive Officer of Foundation Medicine. Brian.
Thanks, Matt, and thanks for everyone for coming. I'm Brian Alexander, CEO of Foundation Medicine. I'm also a neuroradiation oncologist. It's really a privilege to be here today, and this has just been such a wonderful ASCO, and I'm sure we'll hear more about that later. For those of you who don't know about Foundation Medicine, founded in 2010 on the heels of Cancer Genome Atlas and the Human Genome Project, to really take this understanding that cancer is a disease of the genome and really make that kind of available for clinical care, but ultimately indispensable for clinical care. And so, I joined Foundation Medicine in 2018 after the uptake had happened. But when you talk to some of the founders, it just seems such an obvious thing. Like, cancer has been known for 4,000 years, but that it's a disease of the genome over the last 100 and be able to act on that over the last 15. And so, it's been a privilege to start practicing medicine during this era, when the amount of information and the change in practice is happening so quickly. So, it's just a privilege for that. Eric Weiner's opening remarks, if you haven't seen it, they were fantastic. He talked about partnering with patients. We think about this a lot at Foundation Medicine, partnering with doctors and partnering with patients. Eric talked about, it's really privileged as a physician in the oncology space to be able to walk through the door and be able to have that interaction with the patient, that partnership, non-hierarchical, bi-directional. All of us, here that care about cancer have a role to play in, making new advances and getting those advances to patients as quickly as possible. Foundation Medicine plays one role, and, our mission is to transform cancer care, but we can't do that without partnering. And so we really think about that a lot. And so I loved Eric's talk. I loved his framework. I love the theme of ASCO. We partner with doctors, we partner with patients, we partner with biopharmaceutical companies developing new therapies. It's really a privilege to be a part of all of the stuff that's going on. And I'll just give one quick anecdote from one of our patient advocates. She's giving a talk over at our event right now that's ongoing. Her name is Peggy. 2016, she gets diagnosed with a non-small cell lung cancer, just out of the blue, non-smoker, kind of normal family life. And it's just her world changes overnight. She gets a Foundation Medicine test, no actionable results. And so she ends up kind of sitting on chemo, going on chemo, and she, was healthy. And then all of a sudden she's in a chemo chair, kind of angry, confused, given a 5% chance of 5-year survival. So, chemo did well for a while and then she progressed, and, Foundation Medicine recurated her results, didn't run another test, didn't have another assay, but just the information was changing so much for all the work that a lot of you do in the room, that she actually had an opportunity for a clinical trial. She goes on a clinical trial, gets some mileage out of that, but progresses. Another clinical trial, some mileage out of that. 18 months later, her last scan in November is stable. And, the thing that really, her quote that kind of like sticks with me is, just keep me in the game long enough for the science to catch up. And, if you said that 30 years ago, that wouldn't make any sense. But like, it really makes sense in this day and age. And like, for all of us that get to, be a part of this, this evolution of cancer care that's just happening right before our eyes, it's just such a privilege. So I'm really excited to hear all the speakers today and the discussion that's going to happen. Welcome, everybody. Thank you very much for your attention, and I'll give it back to Matt.
All right. Okay, it's hardly worth, I'm going to step up on the stage, but it's hardly worth it because I just have a very quick thing to say. Please welcome to the stage Dr. Richard Pazdur, Director of the Oncology Center of Excellence. Wait, this is going to take a minute. Acting Director, Office of Oncologic Diseases, Center for Drug Evaluation, Offices and Divisions, Food and Drug Administration, and Adam Forrestein, Senior Writer, Biotech at Stat.
My intro is much shorter than yours.
Hey, is this on?
Yep. These are on, right?
My first question.
Yeah, I'm interviewing you.
Hold it. Why do I get the B team? Last time Matt interviewed.
So we're going to start with that.
Yeah.
Oh, this is
So
not,
let's rock and roll, baby.
this is not going well. All right. I'm already spilling water. I'm nervous. I got Rick Pazdur in front of me. All right, Rick, let's get into it. Let's start with, we're going to start with advisory committee meetings. Okay. Rob Califf, FDA commissioner, is your boss. Is he your boss? Is anyone your boss? I don't know. No one's your boss.
Friends.
The guy who's maybe sits
Yeah, we're
a little. Yeah. Okay.
Yeah.
So he's had a few things to say about advisory committee meetings, about the way he wants to change them. I'm going to read you a little quote. We sat down with him a couple of months ago, and he said to us that the most important thing, this is a direct quote, the most important thing to me about advisory committees is that the purpose is not to take a vote on the approval of a product. The purpose to me is to get the advice of experts of various types, whether it's patient perspective, scientific expert, or clinical expert. Almost 100% of people who work at the FDA, when asked, say what they want to know is the reason behind the thinking of an AdCom, not how they voted.
And
Is he right or wrong?
well, as always, there's some nuances. And
No, you have to answer
let me address the
you have to answer my question. Yes or no? Is he
Yes
right
or
or
no?
wrong?
Well,
Votes.
partially right, partially wrong.
Okay.
And let me tell you the situation. Yes.
Swirling out of the answer
Yes.
already.
No, I'm not squirreling. I'm just telling you the truth. Okay. There is a situation, and I've said these exact same words. In fact, I wonder if he copied me on this, because that's my statements that we're interested in why people voted a certain way. And this brings us to two things that Rob had been talking about. Number one, having more meetings that are addressing kind of drug development issues rather than specific drug meetings. In other words, rather talking about a field of drugs or a particular development plan. And one of the things that I've been very disappointed is with some of the companies, when we ask them to come to an ODAC meeting, not to discuss a completed clinical trial, but to discuss a proposed clinical trial, especially if it involved a kind of a novel use of an agent, a novel delivery of an agent. We got a great deal of negative feedback, and it said, no, we don't want to do this. We're fearful that we're going to give away some proprietary information about our plan, et cetera. And so I think that was somewhat disappointing, because I know Rob had really emphasized that in his comments regarding the accelerated, regarding the advisory
we'll get to accelerated approval later. Don't worry.
I know. Okay, so that's one point that I want to make. But the second point that I think we need a vote on here again, we have to make a binary decision at the FDA whether to approve a drug or not to approve a drug. And everybody comes away from an advisory committee with what they heard, okay? And that what they heard is filtered through their own biases, so to speak. So we really need that vote to provide clarity. And I think that that's important. Now, Rob has served as, as the chairman of the cardiovascular ODAC, or the cardiovascular advisory committee, but he's never gone back in the agency, obviously, after a committee to actually have to make that decision, because he really never worked in a review division as such. And a lot of times the advisory committees really think that they know what's going on at the FDA, but they're really not in the FDA itself. It's kind of like people thinking that they know what goes in the pharmaceutical industry, but never work there. There's a much bigger difference. But you really need that binary issue because we have to make the binary issue.
I mean, is that just because we all like to keep score?
No, it means that we have to make a binary decision whether
So you think the vote helps you make that decision, like the
yes.
numeric vote helps you, not just the discussion of the topic.
Yes, it helps us. If we are going in one direction and we hear a unanimous vote that is against what we have been discussing, okay, we have to pause and make a decision. Maybe we're have an inner loop here inside the agency. And this is one of the problems with any regulatory agency or any closed environment, is that you could have a self-feeding discussion, okay? And sometimes you have to step back and say, were we wrong on this? Should we reevaluate this?
So you take the advisory, advisory committee, the advisory part you take seriously.
We take it seriously. And here again, it's the vote, but what goes along with the vote, okay? The decisions and the discussion of what goes along with the vote.
So let me ask you, just inside the FDA. So it sounds like ODAC meetings are going to have votes. If it's a drug review, drug specific ODAC, you are going to have a vote at the end. Is the benefit does the benefit outweigh the risks? Blah blah blah.
Blah 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 blah. Yes. And, you could see this with the members when they're going around the room. A lot of people don't want to make a binary decision. They hem and they haw and they say, oh, this was the most difficult decision. They don't want to disappoint this.
So what do you think about the people who abstain? I hate the people who abstain.
Well, we don't have that many people that abstain. We had one person that abstained in the last ODAC.
Kick them out?
No, I didn't even understand why he abstained, to tell you the truth. I couldn't understand his logic, so to speak. So I'll take it at that, so to speak.
So the basic question is about votes. Like, how do you, because I think people, we do look at the votes and kind of a scorekeeping kind of thing, but like close votes versus votes that are like, obviously if something is like 14-0, we know, right? But like, how do you guys interpret a vote that's like 8-7 or,
Well, it isn't just the vote, then it's the underlying decisions. I will share with this, and here again, I hate to have my back to this side of the room, but one of the things that people don't understand is we have 100 medical oncologists that work at the FDA. And before that ODAC meeting, okay, we have presented this 5 or 6 times to all of this group, okay? I don't know how many times I've heard, well, let me tell you this. I don't know if I've really heard anything at any of these ODAC meetings that we haven't previously discussed, okay? We have gone over these applications in great detail. We've had numerous discussions throughout a 6-month period of time. I probably shouldn't make dogmatic statements that I've never heard something, but
no one's wowing you with their insights at the ODACs?
We have, and here again, we have expertise in disease-specific areas within the FDA. One of the things that I brought out when we started the OCE is to have a breast cancer team, a GU team, hematological malignancy team, people that just do multiple myeloma, people that just do prostate cancer, blah, 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 to reflect what goes on in the academic community. So it's very rare, let's just say that I hear something at an ODAC meeting that we haven't discussed in detail.
So we'll wrap this part up, but
yeah, let's wrap it up.
Yeah. So when you told Caleb you're having votes, he's okay with that?
Yeah, he's not dogmatic. He's not a dogmatic individual by any means.
Let's talk about pragmatic trials. It's a new initiative at the FDA. I know there's one that recently announced it was starting, I think, in April, the NIH.
Yes, let's go over that.
Let's talk about explain to people what is a pragmatic trial and why you're excited about them.
I am very interested because I think it's the melding between what goes on in the real world and the need for randomization. Okay. And what I am very fearful about is with conversations that I see regarding real world data is the feeling that we don't need randomized studies and we don't need randomization. And the issue that I'd like to bring up is that randomization takes into consideration prognostic factors, predictive factors that we don't know about. And that's the whole purpose of randomization. And it would be a travesty if we said, well, let's not do randomized studies anymore. Let's just take this single arm trial and compare it to historical data. So what we're interested in with pragmatic trials is several things. Number one, let's make clinical trials simpler. We have gone overboard in the last 20 years, it's been a progressive continuum of adding things on clinical trials, add this on, add this on, add this, that thing on. So let's make clinical trials simpler, okay? Let's make the eligibility criteria simpler. Let's make data collection simpler, okay? Let's ask the question, what are we interested in a clinical trial? What is the one question that we're interested in? So let me give you the example of how this evolved. Okay, so I'm sitting with the reviewers and we go over clinical trials that we've discussed with different sponsors. And a trial came up of Pembro plus ramucirumab, and it was a small trial in lung cancer. And it really wasn't done for randomization, and we believe it did not fit an appropriate kind of format for a regulatory submission. It did show a survival advantage, though, but we felt it was uninterpretable and it could be due to probably chance. And so what I said, well, we know the safety data of these drugs.
Because these are approved drugs.
These are approved drugs, okay? There's 1 million patients treated with pembrolizumab. Do I need to know more safety data on pembrolizumab? No, okay. So all I'm interested is in one question. What's the survival of these patients? So give me date of randomization to date of death. All I want to see is a survival curve here. That's it. I'm not interested in response rates. I'm not interested in time to progression. I'm only interested in, can we duplicate the survival? So I said, okay, let's redo this trial and let's have the eligibility criteria as simple as possible, okay? What would the doctor do in clinical practice? I'm not interested in, is the platelet count 100,000 or 95,000? If a doctor in real practice treated the patient with a platelet count of 95,000, God bless them, let's put that patient.
So is that where the real world element comes in, though?
That's the real world element, but also the data that is being collected. We don't need data collected on the safety of these drugs. We don't need the response rates. We don't need a lot of x-rays. It's what the dose modifications are basically what somebody would do in clinical practice. So this is kind of the marriage between kind of a real world situation and a clinical trial, with the importance that it preserves the R word, the randomization here. That is what is very important. Now, what we don't know that people have got to understand is when we get a result from this pragmatic trial, okay, how do we put it in the context of a conventional trial? For example, if you see a improvement in overall survival of a month, you might say, well, that's trivial. But here again, that's in the context of this pragmatic trial that has much more noise than a well-done clinical trial. So we're going to have to work with this. But before we get to that, and this is another point I'd like to bring up, many of these trials are going to be over overpowered because we don't know that answer. And we can't talk about the clinical significance unless we have a statistically positive trial. So we're encouraging sponsors to enroll more patients. Once we have a positive trial, then we could discuss the importance, the clinical relevance. But we have to put that in the context that these may be different than the conventional standard trials.
What is industry's response to pragmatic trials?
Disappointingly, I haven't, although we've been endorsing this, I haven't had any takers. Now, we did the first one, and this was my push to do this with the NCI.
And this is the long one we were
This
just
is a
talking
long
about.
one. It doesn't have to be done. I want to make this real clear. It does not have to be done with the NCI. It could be an industry-sponsored study. Now, I've heard, and I've addressed, this. It's, the crazy FDA, but the other regulatory authorities are not going to be interested in this. They want all data collected. So,
You went to Europe, right? Didn't you go to Europe?
I went to Europe. I talked to the EMA on this. I talked to the head of the CHMP, and they said, yeah, we're very interested in this. So then I called up Japan and talked to Dr. Fujiwara, and he said, yeah, Rick, I'm really interested in this. So I think we've really gotten the go here from the major regulatory agencies. Obviously, this is not for every clinical trial. These are for already approved drugs. But I'd also like to point out that there would be kind of a sliding scale here. And let me give you some ideas. For example, you may need more data collection on safety if you don't have a very thorough data package. So instead of collecting data on all patients, you may say, okay, I'll take 20% of the patients and 20% of the institutions and collect the safety data on those patients. Okay. So you could really address with us this sliding scale of where you want to go with data collection. But I really would like to see more interest from industry. And here again, this reflects some of the problems that we've had with industry. We've had a lot of programs that we've talked about, Project Front Runner, many programs, how to do accelerated approval, et cetera. And
The industry doesn't want to do your pragmatic trials. They don't want to do.
well, I can't say they don't.
They don't want to do advisory committees where they're not.
Yeah,
talking
like
about
we could bring a horse to
Oh.
water, but you can't make him drink, baby, right?
Last year you were here with us, and you were a little bit well, not harsh. You were critical of industry in terms of their reliance still on single arm clinical
I
trials.
mean, bitching about single arm trials.
You were a little bit on the warpath about that. I think you called it a pain point, if I remember correctly.
Or a pain in the
1 year later, or a pain in the ass. 1 year later, is that pain been alleviated? What's the current Rick Pazdur thinking about accelerated approvals and single arm trials? And is anyone listening to you, Rick?
Well, the Congress has, let me say that, with their legislation, and I think the community has come out with regards to the fact that they want confirmatory trials ongoing, and that's clear, and I'd like to address that here. One of the things that people don't understand is that, we constantly talk about delays in confirmatory trials, and one of the things that people miss that we see is the reason why we want these confirmatory trials ongoing is not only the delay, but also to answer the question, can the confirmatory trial be done? Because we have seen situations where people promise a trial and the trial is not initiated at the time, and it's not initiated a year later, and it's not initiated 2 years later. And there is a plethora of how should I say it, excuses, including the proverbial dog ate my homework type of thing, so to speak. So, it's a very difficult thing. So you want to have the trial ongoing. Now, that has been explicitly stated. I don't think we could have been much other than getting on top of this John Hancock, what are the Sears Tower?
Willis Tower now.
Okay,
Right.
they changed
The big tall building.
and screaming about this. We said you need to come in with a comprehensive plan. You need to have the trials ongoing, blah, 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 blah. Still, there are people that want to play the system here, okay, and say, oh, we can't do it. We're too small of a company, blah, 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 blah. So we haven't started the trial. Okay. Well, we're going to have the come to Jesus moment, so to speak. And sometimes we're talking potentially about refuse to files on these. But
I think Jesus might be nicer than you.
Okay. Believe me, I went to Catholic school, so
Okay,
don't get into that.
got it.
Baby. Okay. Including a Catholic medical school, so don't get into that. Okay. So the issue is we need to have these trials ongoing. And here again, it's can the trial be done? Okay. The delay is going to be there, but you need to have the trial ongoing. In this issue, we don't have enough money to do these trials. We don't buy that. Okay. Here again, and you know this, the people in this industry better than I do. If the drug was so damn promising, there should be a phalanx of suitors there that would want to invest with you to do these trials. Okay. So that excuse falls
So Rick,
on deaf
what about
ears.
just issuing a refuse to file letter?
Well, that's one thing that we're
Like,
thinking
can't you
about,
just do
but-
that if they don't?
Well, we're discussing that, and I have to bring our other, remember, I'm not operating a vacuum at the FDA. I have to bring the rest of the FDA into Pazdur thing, so to speak. So you have to bring the rest of the agency, but also important is bring the community with us, because I don't want to hear it. Well, this is such of a promising drug, but you're not approving it. But people have got to understand you can't have it both ways. You can't say, well, we need these confirmatory trials. And then when you see a trial that hasn't done it, hasn't started, say, well, forgive those people, because you're going to have two systems here and you can't.
Because you probably get situations where patient groups, advocacy groups come in and are pushing back against that.
Yes, they want these trials. Well, and also bio and other small biopharmaceutical companies that are undercapitalized many times to do these trials. But here again, there's a reason to do it, to have these trials ongoing. The financial risk that these companies are taking, they can't transfer that onto patients. That's simply unfair by not doing these trials in a timely fashion. So I think that is a major point, but we really have to have the community standing behind us on this. You can't have it both ways, demanding that these trials are done in a timely fashion and then saying, oh, we really didn't mean it. We need to get this drug approved. One point that I'd like to say, and it addresses this time to, how should I say it, this refuse to file issue. Once we get a drug into the agency and take it, and I've been stuck in this situation, okay, you take a drug to an advisory committee, a single arm study, a response rate of X%. The clinical trial, the confirmatory trial has not begun. The committee says, yes, approve it. We think the trial should be met the goals for approval, but the confirmatory trial has not been started. It is very difficult for us at that point then to say, we're not approving the drug. We look ridiculous.
You don't have to have a NODAC panel for that, something like that, do you? I mean, like,
no, we don't have to, but usually we would. Okay. Or if
why? Tell me why you would.
For transparency. Yeah. The reasons why we take a drug to ODAC, if you'd like to talk about that, would you?
Well, I'm just saying, like, if
it could go on for like 3 hours.
If they're not doing it, I mean, yeah, you run the risk of a situation like that where you have a positive vote, and then you either have to overrule that.
The problem that we're at, then we're caught in the middle of this most difficult situation, and the risk is all transferred onto the agency for the failure of the pharmaceutical companies not to do the.
I think I would be harder on them than you are. I think you're a softie now.
Oh, no. Huh? Well, okay, you could take the job after I died.
All right.
Okay. Which was the last time we met.
Yeah, are you retiring?
Do we want to take a vote on who's going to outlive who here?
Huh?
We have like 2 minutes left. Are there any questions in the audience? I know I went way too long and forgot to ask Matt to walk around.
No drug specific questions, please.
Please approve my.
Yeah, sure. Thank you so much. This is really informative. You talked about AdComs, Justin Zellen, BTIG. You've been talking about AdComs. Is there in the new PDUFA law a requirement for new drugs to have AdComs? And does that change whether we're going to have more adcoms in the future?
I don't know. I'd have to take a look at the exact legislation on it, but generally we take a drug to an advisory committee if there's internal dispute among the reviewers. Okay, number two, if we want transparency because we are not going to approve the drug, but we're at loggerheads with the sponsor. That's the major issue.
There are other ways to be transparent. You know, EMEA is much more transparent than you guys are.
Give me a break.
Oh, they are. They are. They don't have public adcoms. I mean, they're more transparent than the FDA.
True, in some regards. Okay, they do publish their non-approvals. Okay,
Yeah,
but we're hampered by our laws. But as far as the reason,
work on that.
The reasoning behind that is there, so to speak.
We're going to squeeze in another one.
All right. Thank you. Joel Schindler from Market Vision Research. There is a new entity in Washington, ARPA-H. And one of the big issues with ARPA-H is that it is supposed to play nicely in the sandbox with guys like the FDA. I'm just curious to get your perspective on that, if you've had any interactions and if you have any sense as to how things are going.
They're just beginning. It's really too premature to comment on that.
All right, we had one other here. You've got 10 seconds.
So we were hearing one of those dog ate my homework excuses was people don't want to be in a clinical trial if a drug's already in the market. So the recruitment's very difficult. Do you think that's legitimate?
Well, then they'll have lost equipoise and they shouldn't go on the clinical trial if they already know the answer to the question. So that points to should the trial even be done here again? And that is a serious problem. And we've seen that in many trials where you have a trial where you have differential dropouts at the time of randomization, where somebody finds out that they've gotten the drug that they don't want, for example, and you have 10% of the patients in the control arm drop out because they wanted to get the experimental drug. And that points to a very serious problem of bias. And that bias could not only be in that point of the trial, but also in the interpretation of time to event endpoints of early calling a progression on one arm, later calling a progression on another arm, and has to be looked at very carefully.
And with that, we'll end it. Rick, thanks for sitting down with the B team. Appreciate it.
Next year, the C team.
Rick Pazdur, everyone. Are you going to give me the mic?
It's always nice when we get fresh water. Please welcome back to the stage Dr. Brian Alexander, Chief Executive Officer of Foundation Medicine.
Kind of feel like Congress, where I just want to cede my time back to Adam and Rick.
The audience would probably let you. So I know we had some really insisting questions out there. So, but I'm not sure that your people would be okay if we did that.
Dirty looks.
So welcome, Dr. Alexander. Brian Alexander is the CEO of Foundation Medicine. He's been at Foundation Medicine for 5 years, since 2018. We started as the Senior Vice President of Clinical Development and then became Chief Medical Officer, practiced medicine the whole time until he had this job, which makes you stop, I guess.
I was still practicing, seeing my long-term follow-up patients, which were great, but then, yes, it was getting to be a bit too much.
Before that, he was a neuro-oncologist at the Dana-Farber. And so welcome and thanks for being here.
Thank you.
So, you know, you talked a little bit in your entry about the revolution we're in. We saw some revolutionary data that I think goes right to what you guys do. We had 2 trials, one in glioma, your old specialty, and one in non-small cell lung cancer, the ADAURA data, where we're really dealing with drugs that only work if you can find the right genetic population. Where are we in this revolution? It feels like it's been happening for a long time and it's accelerating, but how fast does it get?
A lot of the stuff that we were brought up just now about how do we, generate information in new ways will determine how fast we're going. Because, if we have to do randomized trials for every single question, it'll take a long time. So I think it's still in the beginning of a lot of the revolution. I mean, a lot of obviously drugs get developed in late stage disease, and then they take some time to move to early stage disease. And the Tagrisso data is amazing and really compelling, practice changing. You know, how long is it going to take for all the other targeted agents in late stage disease to move to earlier stages? You know, there's a lot of questions to be asked. And then the glioma data was really compelling. A drug that works, targeted drug that works in low grade glioma. But you know, to the point about real world data, or in real world experience, how do we use that information? Because like all the patients that are going to get that drug are not going to be exactly like on that trial. So like now in low grade glioma, we know kind of radiation works for progression-free survival, question overall survival, really old data. We know PCV chemotherapy works for overall survival. We know the IDH inhibitor works for overall survival or progression-free survival. And you know, how do you sequence these drugs? If you get a big resection and do you get radiation afterwards, you get radiation followed by PCV chemotherapy. A lot of neuro-oncologists take PCV chemotherapy and give temozolomide instead. When are you going to introduce IDH inhibition? There's just a lot of questions to ask and answer, and those aren't all going to be randomized trials. And people are going to apply the information. Now, the FDA really has a binary decision and really cares about type 1 error to release this into the wild. But then when it gets applied in clinical practice, there's a lot of ways to do that. And so we have lots of questions to answer. The two revolutions that I'm really excited about at Foundation Medicine are the genomic revolution in cancer, obviously, but also the digital revolution in healthcare, where we're able to capture a lot more of the patient journey and learn in new ways. And apply new learnings in new ways. So I totally agree with everything Rick said about single arm trials in general and randomization. But, I think we need to take a really general approach to say, it's all causal inference. Can we understand whether a drug is having an effect and what that effect is having? And so there are instances where single arm trial on an endpoint, we know the drug is the thing causing the endpoint, response rate, those kind of things that are not spontaneously responding. But the question is whether that response is something that we care about as an endpoint for that disease. So we can disentangle the question.
So let's talk about Foundation's role in this. Because you guys have millions of patient report recently, right? A million people have gotten Foundation Medicine reports, and you've launched your Foundation One liquid diagnostic and your ctDNA monitoring test. So how can you guys help this along? What's Foundation's role in making this genomic revolution move faster?
Yeah, I think we have a deep understanding of genomics and cancer. And so we have to partner with others in the ecosystem to generate more evidence in new ways. So that's biopharma partners in trials, ctDNA monitoring. What does it mean? That's another way to look at response, is another way to look at real world data. One of the problems in real world data is there's not a lot of great endpoints that you can look at retrospectively, but is ctDNA, will that be more of an objective endpoint to learn more from patient journeys? So we can work with pharma partners on that, biotech companies on that, academic partners on that. We work very closely with our sister company within Roche, Flatiron Health. So we understand a lot about cancer. They understand a lot about patients with cancer, and that collaboration is really important. We're trying to develop new causal models to extract better insights from observational data, but also develop more pragmatic trial approaches or decentralized trials, but learn more from more patients. Before I came to Foundation Medicine, I was privileged of starting this trial, GBM Agile. And, the really thing behind GBM Agile was that you'd have a patient come in with a glioblastoma, which is a uniformly fatal disease, and you would not have a trial open at the site. And that's just unforgivable. We learn nothing from those patients. They have no ability to go on something new. And so, we just have this ability to be able to be more creative in the way we generate evidence and allow more patients to go on more experimental therapies and learn and give them more opportunities and kind of iterate faster and faster and faster. So those two kind of revolutions, genomic revolution and this kind of novel ability to generate information in new ways. We saw this, we only were able to make inferences on things that were really directly observable, like surgical outcomes for thousands of years. And then, we had hospitals and there was patients all in one place. And there was the Enlightenment and scientific revolution. And we conducted experiments and we learned that way. Now we have to say, this new opportunity with all this information available on patient journey, how do we learn in new ways? Incorporating randomization as a tool for causal inference, but other ways as well.
So what opportunities are you most excited on? Let's talk a little about liquid biopsy. How does that change the game here?
liquid biopsy can change the game in a lot of ways. I think Foundation One Liquid CDX is our liquid profiling assay. And so, obviously it's a convenient way of getting genomic information. We have the same genomic data set panel as we do at our tissue biopsy. We've also worked to say, can you measure the amount of tumor fraction in the blood? And that utility has a couple, there's a couple of utilities for that. One is to say, well, if I don't find any actionable alterations in the blood, is that because I didn't find any tumor DNA in the blood, or is it because I didn't find the alteration? And so we've shown data that if you have a high enough tumor fraction, you can be confident in your negative result, because we've done concordance with our own tissue assay, which is the same gene panel. And if you have a high enough tumor fraction and you get a negative actionable result, you're not going to find anything on tissue. So that's one way to use it. And another way is if you have ctDNA or not, or levels of ctDNA, that's a prognostic indicator. More ctDNA is more aggressive disease. And you can design trials with that in mind and have earlier events and have trials move faster. So liquid biopsy can change the game those two ways. It's also convenient when you can't get tissue. We know that these cancers are going to, the more targeted agents there are available, they're going to evolve over time and have resistance alterations. If you want to track that evolution of the cancer in these new ways, then you're going to not be able to go get tissue every time. So liquid biopsy at those recurrence points are going to really help us understand a lot better this evolution of cancer. And then finally, the ctDNA monitoring, which is kind of closely related to liquid biopsy, is just new ways to understand the cadence of all kinds of diseases in the blood. We've had these markers like prostate cancer, PSA, but now we want to make them more generalizable.
So a lot's changed in the past 5 years since you took this job. I mean, in your old field, the way we deal with brain mets, maybe we saw the way at the beginning of a change in actually the way we treat brain cancers that aren't metastases, right? What do you think happens over the next 5?
I have no idea. like what? I mean, I always say that to that question. I mean, I think one of a great book, a lot of you probably have read, is Emperor of All Maladies. It's just like this terrific articulation of cancer from Egypt to now. And I think it was published in 2009. I don't think it says anything about immunotherapy. Like, it's the most comprehensive, detailed research about cancer. And well, first they say, imagine that you'll be able to go into your doctor's office with a thumb drive of all the genomics on your cancer, and that'll be like 2050 or something like that. Like, that's now. And then, immunotherapies have changed everything. Now, how many other treatment modalities are there out there? So, we just have to keep an open mind and learn as fast as we can. And, all the discussion that you just had with Rick about how to really take information and learn from it in new ways. And really, we should look at the same set of information and have wildly different conclusion about what it says. So can we quantify uncertainty? Can we quantify how people are making decisions? I think those are all the things that we're going to need to do in the next 5 years.
All right. A quick final thought.
No, just again, thank you everyone for coming. I mean, like, a lot of gratitude. I mean, I feel, to your point before, when I started, I started my clinical practice in 2010, and I took my oral boards for radiation oncology in 2010. I'm a CNS radiation oncologist. If you would have said at that time, any patient with greater than 4 brain mets, you don't give whole brain radiation therapy, you would fail your boards. And like, within a couple of years of me starting practice, when these drugs start coming out, the targeted agents, and you've like 20 brain mets, and you're just trying, well, maybe we should just hold off and see what happens. It's a big leap, but that's kind of standard now. And so things are changing so much. There's so much information being generated. It's such an exciting time to be there. Again, back to the Emperor of All Maladies, 4,000 years, there's like nothing but surgery. In the last 100, you get kind of radiation chemotherapy. In the last 10, it's like exponentially growing. So it's just such an exciting time to be in the oncology space. If you care about cancer, you care about people living with cancer, you really feel like something is happening every year, and it's just like such an exciting time and a privilege to be there.
Wonderful. Let's all take some hope in betting in exponential growth. Thank you so much, Brian.
Appreciate it.
And I'm staying here, but so it's been a big day for our next speaker. Could we please welcome to the stage Dr. Susan Galbraith, the Executive Vice President of Oncology R&D at AstraZeneca. Thank you, Susan. So it's been a big day. How was your day?
It's been a great day. Thank you.
Been a good one?
Yeah.
So
I'll take going to a plenary at ASCO for anything.
Take going to a plenary at ASCO. Was it a good? Was the applause as much as you hoped? Were you?
Yeah, no, I think that both the audience and the discussant had a good appreciation of what the data mean.
So one of the difficulties for me of this interview is that Susan's actually relatively humble, especially for people in our industry. But I wanted to, you're wearing a pin.
I am wearing a pin.
What does the pin say? Everyone, I don't know that we can zoom in on it, but it's got an AstraZeneca symbol and then there's a number next to it.
The number is 5.
And what's that number mean?
It's the 5th year in a row that we've had a plenary at ASCO. So there was.
And those were Lynparza.
It was Polo, pancreatic cancer.
Mm-hmm.
It was, Adora with the disease-free survival data.
The first data from.
There was Olympia, which was for adjuvant breast cancer for olaparib. There was Destiny Breast 04.
In HER2?
With standing ovation. And there was the Adora second time around with the overall survival.
That's a pretty good run. So you've been at AstraZeneca since 2010.
I have, yes.
In 2010, I think people would have been very surprised at the idea of AstraZeneca having 5 plenaries in a row.
Yes.
Just taking us back there, that was the year that Iressa was finally completely withdrawn from the US market. That was AstraZeneca's first breakthrough cancer drug, which was removed and is really the predecessor of the drug we saw today.
Yes. So it's organizations go through, iterations of how they're organized. And that time at 2010 was a big change for AstraZeneca, where the whole of the R&D was redesigned. I mean, you're right to point out that Iressa is the predecessor of Tagrisso. We have Tagrisso because we had Iressa, and that's a really informative element, because it speaks to the fact that you need a degree of persistence in understanding the cancer biology. So at the time that Iressa was developed, we didn't understand about EGFR mutations within lung cancer, and so the drug was developed in an all-comer patient population, not understanding that cancer biology, because the great genomic revolution that Brian's Alexander's talked about wasn't there to help inform that piece. And the information that about why some patients that were treated with Iressa were having particularly impressive responses came out during the development of that drug, and subsequently via the IPAS study. It was tested in the patient population that was more likely to respond. But during that whole learning journey, first of all, there were a few things. AstraZeneca chemistry group had learned a lot about kinases and how to drug kinases with more specific inhibitors. And a lot of that know-how and knowledge was within the company and within both the compound collection, but also the heads of chemists that had gone through that, because there were several kinase programs that we'd had. Secondly, we'd established relationships around the world with investigators that were used to treating now what they knew to be EGFR mutated
places like Asia, where there was a lot of EGFR and the drug
exactly
was better.
From Japan and South Korea and China, and those relationships are things that enabled us to go quickly with Tagrisso. So when we started the Tagrisso phase 1 trial, it was called AZD9291. At that point, we started it in parallel in Asia and the West, and that enabled us to go quickly. We started it with a test for the T790M mutation, which we knew about because we had relationships with investigators, and so there was a reason why Tagrisso was a very fast clinical development, because it was built on the knowledge of things that had learned. And so
And you weren't in your current role then, but you were involved in this decision. One of your first jobs was to do a portfolio review, and you told me previously that you saw this program and you felt it had a higher probability of success, and you put everything on it.
We did. It was in lead optimization at that point. So the actual program was started in 2009. I arrived in 2010, and within a few weeks had to, cut a certain proportion of the pipeline, and then choose where we're going to prioritize. So this is one of the programs that we put a bet on. We put a lot of chemistry on. We put a lot of effort into so that we could accelerate it. It's a competitive space. We didn't know how competitive at that point. You never do when you're in that stage of development. But we moved it at pace, and that really has made a difference for that development.
Could we get our clock up here? It's stopped. It'll be right once, but so another compound that was floating around at that time was olaparib. the other one that you mentioned in a bunch of those plenaries that you've had.
Yes.
Um,
Another story.
Lynparza. Yes. And you've said the story of olaparib is a story of comebacks.
Yes.
But it was a long and winding road. You got rejected at an AdCom. We were just talking about Rick before you could get this drug approved.
That is true. Yes, it's not the setback; it's the comeback. What they said.
But you said that was one of your lowest moments in 13 years here.
Well, because at that point we knew about the patient population where olaparib was likely to work in BRCA mutated patients. In this case, the initial trial was in patients with ovarian cancer. And the story of that trial, the Study 19 trial, is an interesting one because it was a phase 2 randomized phase 2 study. In the maintenance setting in platinum-sensitive relapsed ovarian cancer, but still relatively small. And at the time when that trial first read out, the Study 19 trial, we didn't have the BRCA mutation status on all of the patients. We had it in about 20% of the patients. And I saw something that I've never seen before. I saw a hazard ratio in a clinical trial with a point estimate of the hazard ratio of 0.17. Now that wasn't the p-value, that was the hazard ratio. And so the overall PFS benefit was in the range of 4 or 5 months, which is good, but not Q-SOF brilliant outcome. And so but you had this intriguing finding, and at this point in the company's history, people were questioning whether or not we were actually going to be able to get approval without overall survival. Whether we were going to get equivalence with, so with a capsule formulation at that point, whether you're going to get bioequivalence with a tablet formulation and what that was going to mean. And, there were questions that were leading to questions about whether we should continue to be investing in this program. And so I argued for the investment to do the testing on the remaining patient samples that we had to see whether the remaining patients, for the 80% that we hadn't tested, when in those patients, if you had a BRCA mutation, you'd still continue to see this hazard ratio. And it was about $1.5 million, if I remember, to do that. So I do remember saying that it was one of the best investments that I've argued for, because it helped to get the data. And yes, the hazard ratio was recapitulated in that patient population. And ultimately, we have taken that forward into the SOLO 2 trial, into the SOLO 1 trial in ovarian cancer, and shown the benefit in that patient population for this kind of approach. So yeah, it was about really understanding, again, the right patients to treat, taking it forward, and then taking it to multiple different cancers, as you say, not just ovarian cancer, but breast cancer, into prostate cancer, and also into the pancreatic cancer, the POLO study.
So a blockbuster drug in several different diseases. That around the time, that was 2014 that AdCom happened, and the first patient was dosed when for Tagrisso? It was 2013.
The first patient went in March 2013. Yeah,
March 2013. So something else changed around them, which is there was a new CEO who joined, Pascal Soriot, who is still the CEO. Mm-hmm. How did the culture change when Pascal joined?
Well, completely, because what he said is we follow the science, and the science is at the forefront of the decision making. So the whole, philosophy of the company, science had always been important, but it was put as the central pillar, and everybody needed to understand that you start with that as in the decision making. So that I think that's a fundamental shift in culture.
You told me that your interactions with your commercial partners changed within AstraZeneca.
Absolutely. And so, the commercial team have also got to be steeped in the science. That was the point that Pascal was made. They've got to understand, be steeped in it, and be familiar with it. And what we now have is a collaboration across R&D and commercial, where we're really we are collaborators together, and there's an interaction. So we learn from the feedback that we get from the investigators and doctors that are treating patients once the drug is approved. That's fed back more rapidly, but also what we're doing within R&D is is fed forward into what the how the commercial team's thinking about it, how to understand which are the right patients to treat, what the meaning of it from a clinical meaningfulness of what you're trying to show is going to be. And I really think, I mean, I've been in the industry now over 20 years. That level of connectedness from the early part of discovery through late phase development and into the commercial thing is an unusually good collaboration. And I think that's part of the secret sauce of what we've got at AstraZeneca.
Any questions out there for Susan? Fierce Pharma here.
Angus Liu from Fierce Pharma. So Susan, at that advisory committee meeting for Lynparza, obviously FDA was questioning that had we designed this trial differently, we would prospectively understand some of the subgroups analysis. And then right now we have dual the clinical trial where you have the primary investigator once again saying that had we designed this trial previously, we might have a Lynparza, separate Lynparza arm to kind of tease out the contribution of different agents. So I think we've entered this place where we have the cancer drug development field is just moving so fast that when you read out a clinical trial, that it might not be relevant anymore. So how do you deal with that kind of pace of drug development to incorporate into your design of clinical trial?
Yeah, so I think first of all, just to clarify, I think you're talking about a different ODAC for olaparib. You're talking about the prostate cancer ODAC, not the one that Matt was talking about, which was the original one for ovarian cancer. Just to be clear. So, but I think the principle that you're also raising is at the time you design the trial, you know less than at the time that the trial reads out. And over the 2 or 3 years that you can be from the design point to that readout, the knowledge base can change, particularly about what the biomarkers might mean and which are the patient populations that might be particularly like. So that is a true aspect of that. Now, you work very hard at the beginning part of the trial design, and this is a place where real-world evidence and data are incredibly valuable to try and understand better which are the patient populations and how they perform on the current control arm better. And so with the advent of connected data sets with real-world data that's got genomic data and other diagnostic tools linked to the outcomes of patients on real world, I think that's helping us to actually drive those designs in a more effective way. We embed it now into all of the phase 3 designs that we do to help improve that. And we have a collaboration with Tempus, that is very helpful in that regard. But we obviously use Flatiron data as well. We use Flatiron data as well. So we use real world evidence. What we need is real world evidence with that quality of data globally. Not just in the United States, but from multiple different regions in multiple different areas, because the information can differ. So I think that's one thing that can really help. And the second thing is that, when you design your trial and, as you're going along, potentially, modifying your statistical analysis plan, you can take into account sometimes some of the changes that are happening during that. Specifically to your question about, you were referring to the DUO data that was presented earlier at ASCO. Of course, when you cross-compare with parallel, it's got all the caveats of cross-trial comparisons, but I still think you can and we will probably at some point show matched analysis data so that you can help do that kind of cross-comparison. So watch this.
Susan, I want to ask about another, I asked you about Pascal, but as time winds down, I want to ask about another person you worked very closely with, which is Jose Baselga, who had your job before you, but before that was an advisor to you on olaparib and some other programs, and was, I think, very involved in bringing in HER2. I kind of want to ask this as one question, which is, what was it like to have someone like that working with you, but also what was it like to lose them? Because you lost him very tragically just a couple years in.
So I'd known Jose for many, many years, going back to the time when I was working at Bristol Myers Squibb, when he was involved in different advisor we was then. So I've known him and counted him as a colleague and a friend for many years. I mean, I've told this story about we actually went skiing on time in Utah, and he took me down this ridiculous, because he's a better skier and his wife's an even better skier. It's been down this ridiculous slope. It's a triple black diamond, which I think is just bragging for a slope, really. But it was a narrow tree chute, and it was basically one of these you turn or die. And he sort of looked at me, giving me a little grin, and said, "Just follow my skis. Just follow the back of my skis." And it was like, it's not an option. You know, you just do this. So Jose was a magnetic personality and a compelling individual, absolutely passionate about developing drugs to treat cancer and developing the strategy. He joined AstraZeneca because he thought that the strategy that we had laid out made a ton of sense and was very aligned with what he wanted to go. So it was a great fun time co-developing the strategies that we've played out. And so yes, to have somebody of that magnitude, a giant of cancer care, lost to a dreadful disease like Creutzfeldt had an impact on all of us. But you know, the organizations that we work in are always bigger than any one individual. And so no matter how big, bigger than me, bigger than Jose, bigger than Pascal, bigger than everybody, we work together as a team, and it's the team that matters. And what I was incredibly proud of is that the team pulled together during what was a difficult period, and we did not miss a beat. We did not miss a beat, and it was great. And it was something that Jose would have been very proud of.
What did it feel like to take over the role after that?
Well, you know, I know AstraZeneca well. I know the team well. I know the drugs very well. We had a partnership, and so it was in some ways a natural evolution to take on that role and to drive it and be committed and deliver against the vision that we had created.
Do we have another question out there? No, because I'll have to close. And so okay, so we've talked about 5 ASCOs in a row. I also, another stat I didn't mention is how many drugs in? So
we've managed to take to approval 8 new drugs since 2014.
So what's next? There's
so much next. So of course, I think we continue to the journey with Tagrisso and with Lynparza. There are more trials that are ongoing. There are more readouts. We just announced high-level results for DUO-E, but which will get presented at a congress later this year, we hope, which is an endometrial cancer, again, a combination with Imfinzi. We've also just this last week also announced that we've got high-level results for the pathologic complete response rate for early stage gastric cancer with Imfinzi in the Matterhorn study. So there's lots more that are coming through. And then I think what you will see even later this week with the Destiny pan-tumor data is that drug like Enhertu has potential beyond what we've seen already in the settings where it's already approved in breast cancer, in gastric cancer, in lung cancer, in multiple different tumor types. So we're looking forward to sharing those data. And I think that will generate ideas about other things that we can do. I do think that antibody drug conjugates, as we now constructed them, have the opportunity to be better than chemotherapy standard of care in many different settings with many different constructs. And we've got some of our own coming through into the portfolio that we're excited about. We've also got, I think, ways of thinking about how we can improve on current immunotherapy. We've got a number of bispecific molecules, PD-1 CTLA-4, PD-1 TIGIT, and PD-1 TIM-3 that are in development, but also we're investing in the immune cell engagers and cell therapy. And I do think that cell therapy for solid tumors is going to become a reality. And then the other aspect of our strategy, not just to go early, not just to drive in combinations, but to pull together an integration of this data and AI, better diagnostic testing, including a revolution in digital pathology with computational oncology tools that are going to be needed for the multiple ADCs that we've got, but also digital health technology so that we can have more patients spend more of their time where they want to be, at home with their families. We can potentially mitigate the onset of adverse events by identifying them earlier and more truly patient-centric care. If you integrate all of that vision together, I think there's more transformations that are just around the corner. So I think it's an incredibly exciting time. I think Brian Alexander is absolutely right. It's an incredibly exciting time to be in oncology, and a privilege to work with a fabulous team and the whole ecosystem that are helping to change how cancer is treated. It's a golden age.
All right. Well, that's a golden spot to stop. So thank you so much for being here. Congratulations on 5. We'll all see if you can pull off 6.
No pressure. Thank you.
No pressure. Never. All right. I'd like to welcome our last panel up to the stage. Thank you. I'm stuck up here. So I think there are going to be some chairs coming up first. And then I'd like to welcome to the stage Angus Chen, cancer reporter at STAT. And welcome back, Adam Feuerstein. You know Adam. And I'd like you all to meet Catherine McPhail, who will moderate. She is programmer editorial events at STAT. You go there. Yeah. Sit down.
Did everyone get their sunglasses?
We should have really all had them so we could do the Blues Brothers thing right now. And we are in Chicago.
True.
Well, Adam's wearing shades and has a pack of cigarettes and
No, I don't have that.
yeah.
Sorry.
Or whoever that line goes that I used to know. So just before Catherine goes, interrupt us, throw out questions. It'll be fun. Please ask questions.
Yes. Is this on? Okay. Well, to kick us off, we were just hearing about Tagrisso from AstraZeneca for non-small cell lung cancer. Matt, this is being called paradigm shifting. Could you tell us why that is and why this is some of the most major data coming out of the meeting?
I mean, 0.49, half, right? I mean, when you see an OS curve where you're changing the curves like that, it's just a big deal. Angus, I mean, you talked to a lot of people about this.
I did, yeah. I think that, well, AstraZeneca had a press release earlier saying like, hey, like our OS data is significant. We didn't have the numbers at that time, but we reported a little bit. When the numbers did come out, I think people were like, okay, this is a big hazard ratio. It's really nice. And I think what it said to a lot of people was, we were like, you know, the oncologists who were like starting to treat with Tagrisso in the adjuvant setting were like, oh my God, like we were validated it in believing the DFS data, which had like as you most of you probably know, had an incredible hazard ratio. It's an 80% like approval, I think. And I think that also is leading some researchers to think, well, you know, actually maybe we can use DFS, or maybe we can use even earlier endpoints for starting to test drugs for rarer aberrations, like something like NTRK, where you could never accrue a trial that would use overall survival as an endpoint. It would just take too long. I think one researcher said like that trial would probably read out at my retirement. So you still want to have these drugs, or you still want to like study these drugs, but then like, you know, OS is the gold standard. How do you actually get that? Maybe you'll never get that. Maybe we'll never be able to test these drugs. I think with Adora, people are saying now we can shift that paradigm a little bit. Maybe we can start to think, hey, some of these earlier endpoints might be enough. I feel like I think it's tricky, of course, but
I do feel like, if we step back, this was a pretty good ASCO so far. I mean, obviously there's a couple more days, but for overall survival, I mean, there was, you know, the study we're just talking about, and then the Ella Hare study in ovarian cancer. Tomorrow there'll be results from Zuma-7, which is the Yescarta study in lymphoma. We know that the study was positive. The data specifics will be out tomorrow, but again, another survival study. So it's been like a pretty good, big studies, survival benefit kind of ASCO.
But we also have progression-free survival with the vorasidenib data in glioma, in an indication where I think survival is going to be really hard. And I think we're going to have some interesting ORR data coming out, overall response rate of just shrinkage. So, you know, it does seem to be sort of case by case. I don't know that I see Rick, who was here, letting go of overall survival that easily for the NTRK and ROS and ALK and all those wonderful drugs there.
But the other thing, interesting thing about the Adora study, that sort of the whole, if you remember a couple of years ago when the disease-free survival data came out, there was a lot of controversy about that. Jack West, sort of famously, West Coast lung cancer expert, who was like kind of crapping on the data a little bit, and like casting doubts about it. And there's a lot of, if you were on Twitter, there was a lot of lung cancer docs who were going back and forth about it. So it was there was a little bit of this time of like, the guys who were, Roy Herbst was obviously a big champion of it. A little bit of like, I told you so, I think.
I think he was definitely a little bit like, I told you so. He didn't quite want to say it. Even Susan Galbraith, I remember, I talked to her and she was like, is it vindication? Maybe.
This was also a bit of a coming of age moment for antibody drug conjugates, such as the Elahere drug from Immunogen for advanced ovarian cancer. Adam, could you tell us a little bit more about
Yeah, I mean, those are pretty, the whole field of antibody drug conjugates is, it's increasingly popular. Obviously, the Pfizer deal to buy Seagen was all about ADCs as a sort of a backdrop. And so, right, Elahere is a ADC for ovarian cancer that was Rick Pazdur's not here, but this is they got an accelerated approval for late stage ovarian cancer, and then ran a confirmatory study with survival, PFS and survival as endpoints, and hit on both, showing a survival benefit in a randomized study. So Rick hasn't seen those confirmatory data yet, but when they will be submitted, and I assume that it will get full approval. So that sort of, does what he wants to see happen in these kinds of situations. And, obviously it's a meaningful, the words practice changing get thrown around a lot. It gets to be a little bit almost like, I don't want to say cliche, but, lots of things get called practice changing. I did speak to physicians who treat women with ovarian cancer, and they were pretty impressed with the benefit that was observed in that study. And it was very meaningful.
Do we have any audience questions at this time? Oh, we have a mic coming.
So Matt, you were at the same press briefing I was at the other day, and I can't remember the details at all, but there was a study, and it was not of a new drug. It was of like using an existing drug, but using it in place of radiation or in place of chemotherapy. The point of the study was like, people, we don't need to, like, this is not just additive. We don't need to use as much radiation, as much chemo anymore. And they talked about how kind of like, that's one of the themes that they're seeing at the show is these new oral drugs, not just as additive, but like, can we get away from some of the toxicity and some of the sort of like harmful treatments?
Are you talking about the glioma results, the lymphoma results, or do you not remember?
I guess I don't remember. No, I think this was the lymphoma.
Okay.
Angus
has thoughts on this, I think.
Yeah,
I do. I mean, I think I almost thought your question was a little tongue in cheek, because it is sort of all these things that, at least since I've been reporting on cancer, I've sort of noticed that a lot of researchers and clinicians have sort of been backing off a little bit on just throwing all the guns at, like, even patients that have like very good responses, even like low risk disease, early stage disease sometimes. And it's not just about like better drugs, I think, always, because I remember covering a story months ago, looking at low risk thyroid cancer. And for thyroid cancer, sometimes patients will take radioactive iodine orally to try to like concentrate that radiation in their thyroid and treat it. And some of these studies were showing like, actually, you might not need to do that. You just your standard care is going to be fine. You don't have to also add on the radioactive iodine. And so I think there's just a general movement towards like, maybe we hold off. Maybe we don't like do everything possible to these patients that we can do. But I think that the newer drugs, sort of newer agents, better drugs, that does have a lot to do with it, because you can get like better responses, more like complete responses more often. And that's great. And if you don't have to, irradiate healthy tissue, then by all means, I'm sure the patients will appreciate that.
this is the long arc of curing cancer, right? Like we started with giving terrible drugs to little kids. That's how we started curing cancer. You know, mustard, right? Like, I mean, and we've seen it in a lot of, I mean, the move decades ago away from radical mastectomy was a good example. And, you know, look at HPV cancer and people who think that maybe we don't need to treat that HPV head and neck nearly as aggressively as head and necks traditionally. So I don't think it's surprising, but I do, Angus had noted that there have been a bunch of these radiation sparing studies, which is a nice thing to see.
Kevin.
Thank you. Julio Heidenberg. I work at the VA in Boston. I'm a hematologist and oncologist, and I know all the discussion about OS and PFS, but something I've been emphasizing to our fellows is the value of PFS, the time that you gain with drugs that extend your time until you get whatever you need to get, because the value of 3 years today, given the pace of innovation, is much higher than the value of 3 years 5 years ago. I mean, just look at this ASCO compared to last year's ASCO or before the pandemic ASCO. And so it's just tremendous, the amount of exponential change we're seeing. So I have, and I also tell them that whatever you learn in your fellowship, the half-life of what you learn, it's obsolete in 4 years. 50% of what you learn is not very useful. So we have to teach you how to learn and refresh yourself all the time. I was going over the massive amount of information, and you know, you talk about these things of journalists, but oncologists, we're having a big problem unless you're a subspecialized person grasping all this stuff. I know this is very important for survival, for PFS, for quality of life. You know, the rectal study that was presented today was impressive. It took 10 years. By the way, right? So there's a lot of stuff going on. But anyway, I wanted to emphasize the issue of the PFS. You know, the value of 3 years today is a lot more than 3 years ago.
And the value of time to patients. Getting the next, getting to the next drug.
And there's going to be more drugs in the next 3 years than in the past 3 years.
And the value of journalism, right? Like read stat will keep you up to date.
Nice plug.
Thank you.
Our boss will be happy with you.
Matt, there was also new data, like you mentioned, for vorasidenib, a drug from Servier Pharmaceuticals for brain cancer. Could you tell us a little bit about that data and what was interesting about it?
I'd heard a rumor, and it may not actually be true, and I don't see him, but is David here? No? Yes, David, are you here? No? David Shankine, who was the CEO of a company called Agios, which developed this drug, which was then sold to Servier. It's just kind of a crazy biotech story. And now we're looking at what were really impressive data. I would really love it if this drug hadn't had the liver tox issue. It may be, I'd love to hear if people out there have an opinion. Usually hearing the words Hy's Law in the context of a drug, you know, in my cardiology world where I've spent a lot of time, I would write a story saying, well, this one's not going to get approved, but it's obviously different in glioma. But the efficacy was very positive, and it really points to using IDH inhibitors in this disease, you know, seeing a 60% reduction in growth in a disease like that and giving people, again, more time, and there hadn't been any progress in decades. So, I mean, it was a great thing to see. You know, it'll be very interesting to watch the regulatory review of this one, even though it's not a company that people can invest in. I think everybody who's watching targeted therapy should pay attention to this one.
I think we have an audience question. Yep.
Hi, Shivani Vora, AZ. I have a question, just generally speaking. So obviously this ASCO was great. We saw a lot of amazing data from each of your perspective. What do you think was missing or we could have seen more of just from an overall oncology space? Thank you.
In terms of data or missing from cancer care, which I could answer much more easily. So okay. How about, I mean, we heard a little bit about GRAIL, but like better screening, and we don't use lung cancer screening enough now. You know, we're getting all these results in adjuvant lung cancer. We treat a lot more adjuvant lung cancer if we could find the lung cancer earlier.
Yeah, that was a point that came up. I went to see the presentation of the perioperative study in lung, immunotherapy, but again, using it before surgery and after surgery and getting patients, in the earliest stages. And the presenter made the point that, what this does is this calls out for, we're only going to be able to get those patients if we screen, right? And obviously for unfortunately, far too often in lung cancer and other cancers, we don't catch, or people don't get diagnosed until, the disease is much more advanced. And so to leverage a lot of the stuff that we're doing, that the industry is doing and researchers are doing now, is we need more screening. Yeah, for sure.
I would say I don't think this is completely missing from ASCO, because I do see it pretty much every year. But I think health services is really important. Like, it's great to talk about what's on the cutting edge of research in cancer medicine. But at the same time, how are patients actually accessing these medicines, and how are they interacting with our health system in a way that's going to be effective and efficient? Because still right now, so many patients in the US and around the world don't have good access to a lot of essential medicines. And a lot of it just comes down to health services and how these systems work. I think cancer researchers and clinicians think deeply and put that more on as a priority, not just what are the best, newest medicines, but how are patients actually going to be receiving them?
And we need more Taylor Swift concerts too, right?
Yes.
There's less discussion of financial toxicity I hear this year, but people's out-of-pockets are going up.
Yeah, absolutely. Yeah.
So,
sorry, go ahead.
Okay, Xiaolin Liao from Immunotherapy Therapeutics. I would like to advocate for screening for gastric cancer. Gastric cancer affects a lot of people from Asian and other developing countries. And it's very easy to screen for H. pylori. It really should be a very universal and simple screening. And then I'm very impressed with all the data presented at ASCO and the doctor's comments on, 3 years progression-free survival means so much for patients because of the speed of innovation, right? So for this panel, you interface with the industry, with the inventors, investors as well. Are you worried about the 3 years lost due to the lack of investment in innovation with the current biotech situation and the financing situation?
Absolutely. Although I'm also surprised how many big rounds there still are in the private market.
I don't know whether there's a lack of investment in cancer research. When I speak to investors, what I hear often, the biggest complaint that I hear from investors, people who fund companies or invest in cancer companies, is that cancer is a very difficult field to invest in. It's very crowded. You got a lot of companies all chasing after the same targets, and it's very hard to kind of separate out who has got something that's real versus not. And so I think a lot of investors sometimes look elsewhere outside of cancer. And I think part of that is, and part of that is also the consolidation that, a lot of pharma companies, these meetings have become increasingly big pharma meetings. We had AstraZeneca up here,
Merck,
where,
Novartis.
yes, there are always going to be little companies, smaller companies that are going to be developing things and they get acquired. I saw some data this morning from a Chinese company that was, that licensed the drug to Merck, and actually an antibody drug conjugate. They had some really promising data this morning. So I think you're always going to see those kinds of deals, but it is becoming increasingly like a big pharma world.
I also think that oncology has been the dominant recipient of biotech investment for quite a while. I mean, you look at a large share of the number of drugs in development for oncology, and we finally had some progress in other areas. Like there's a lot happening in diabetes all of a sudden. There's the GLP-1s, which you can't stop hearing about in obesity and potentially in CV disease, but there are other drugs there too. And so it may also be a case of how much of the pie cancer gets shifting. And we want to cure cancer, but we want to cure cardiovascular disease too. So that would be my other, but yeah, I worry about, but also some of the best biotech companies and some of the best drugs come from periods when things go fallow. It's not the case that if investment doesn't just keep going up, 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 that innovation won't happen. We haven't figured out why innovation happens. So it's not great when it goes down, but it's not always the end of the world.
So I'm going to ask you a question. You know, you guys are journalists, and how do you draw the line? You know, we saw the leak of commands and Cartitude-4 way before this conference. How do you guys, and I don't know whether Stat actually reported on those leaks or not. How do you guys as journalists?
Well, I mean, those leaks, I mean, let's
and I patted him on the back, just to be clear.
No, but those leaks were mistakes made by, well, in those cases, it was made by EHA.
Okay.
I mean, so it wasn't like somebody was like stealing the data and putting it on the internet. There was like a web problem, and the data got out there. So we reported on some of it.
So
because it's out there.
As journalists, how do you guys internally draw the line between leaks, data, mistakes, somebody's leaking something, like, you know, what gets reported, what doesn't? You know, I was asked about both commands and Cartitude-4 for like a month before I even came into ASCO, and it was like, I
didn't know that. Well, all right. So I think what he's talking about, he's referred to, and I don't know if everyone is familiar. So EHA, which is the European European Hematological Association, right? It's next week's conference. It's like the ASH equivalent in Europe. So they have their website. They screwed up, and abstracts that were supposed to be embargoed ended up on the internet. Somebody found them. I don't know who, but somebody found them, and then they spread around. And so, you know, it's all sort of fair game at that point. I mean, it's out there. And so we, like, for instance, when the Carvykti data from Cartitude-4, which is the BCMA targeted CAR T, those data got out. Yeah, I wrote a story about it because the data are out there. I mean, what am I going to do about it? I mean, I didn't leak the data, and nobody leaked it to me secretly. It was like on the internet. And so it's out there, and things were happening. And, I called to confirm. I mean, I did call, I called J&J, for instance, and to make sure that the data were real and not made up. Right. I mean, that was the professional journalism thing to do. And I did that. And they said, yeah, unfortunately, those leaked, and yeah, those are the data. So, okay, then we write about it. Stuff like that happens, you know, but well, I mean,
if we can confirm it, we probably report it.
Yeah.
I mean, so we as journalists as a profession, I think we generally have an ethical belief in helping information be surfaced. There are certainly exceptions. There are certainly things I've learned that I've decided not to report for various reasons. It's not something that belongs in the public domain. It's too personal. But we do generally believe, and in the case you're talking about there, I mean, I can kind of hear Adam for 20 years here talking about, you've got small investors who aren't getting the information, and large ones who are, that you're often not keeping information private. You're often creating asymmetries of information.
Let me put it this way: we're not stealing data.
Right.
Well, but I'm saying, we're saying, where do you draw the line? Right. I mean, I'm not going to go out and steal something, but if somebody has something, it's just there. I mean, right. Sure. Yeah. Right. Well, yeah, I mean, again, that's probably an unfortunate example where, but I don't think that happens all that often, does it? Do you think? I don't think it does. I mean,
it's the job of the people with the secret to keep the secret. If you're telling a journalist, you're not keeping the secret.
We're not ones to keep secrets.
It's our, if
right?
you tell, just everyone knows, we will talk to you on background.
We have like
gladly here at this
that
event.
don't allow us to do that. Like,
but if you tell us something on background, our goal is to put that on foreground.
I mean, I always say to people like, what's a journalist's job? It's to get information that we're not supposed to have and then tell everybody about it. I mean, that's essentially what our job is.
We have other jobs too.
We
explain stuff.
Are just about at time, and we were talking about access to care earlier, and that's actually a note I would love to end on. So Angus, I was wondering if you could close us out by telling us about the twinning initiative and how that helped improve health outcomes.
Sure. I'll try to be as brief as possible since I know we're at the end. But this is a program that St. Jude's does with a lot of hospitals around the world, but specifically an abstract was presented at ASCO, I think yesterday, that looked at a twinning program between Rady Children's Hospital in San Diego and Hospital General Tijuana. And I thought this study was really interesting. Hat tip to Matt, who pointed it out. And the idea of these programs is to sort of create sister hospitals that will share training resources, what else? Like just expertise, and sort of just anything, almost anything you can think of. It's hard to say in just a few words, but it's basically like building infrastructure almost on both sides. But the idea is to benefit low and middle income countries or hospitals in low and middle income countries to sort of improve the quality of care. And Adam, you're talking about survival being a huge thing at this conference. What was cool about this was after this program was implemented, and they were specifically looking at pediatric ALL. In 2008, before this program, survival for pediatric ALL in Tijuana was 10%. And then after it was implemented, it went all the way up to 65%. And that's crazy. If you could find a drug that can do that, that would be completely insane. But this wasn't a drug. This was just making sure that children have access to the drugs we already have and the best protocols that we've already set. And I think that's not a difficult thing to do. You could do that in almost every hospital in the world and every hospital in this country, because a lot of rural hospitals in the US don't always have access to or I guess a lot of rural areas in the US don't always have access to the best cancer centers. If you live in Wyoming, for example, you have to go to another state if you have pediatric ALL. And so I think there was a lot of lessons in there on how we can improve access to care, how we can just do things to massively change survival for cancers that we know a lot about without ever inventing a new drug.
So let's end this on kids not dying.
If anyone has any information they want to leak, just meet me out back.
You can tell me too.
Yes, me as well.
Angus is also here.
Yes.
You can tell any of us your information you want to leak. Oh, this is awesome. So I'm going to give my closing remarks now. These guys can get off stage. They can just stay here. There are drinks outside still. We'll be open for another hour. I want to make a few. I want to just tell you a little bit about the summits we'll be running late in the fall. So the Future Summit, which is our virtual summit in September, with George Church, Jerry Mendel, one of the biggest names in muscular dystrophy research, Dr. Robert Wynn from the Massey Cancer Center, Ugur Sahin from BioNTech, Vin Gupta from Amazon, and Brian Wallach and Sandra Abreev from IMALS. I promise this is going to be 3 days. It's going to be a few hours each day. It'll be a really cool way to use Zoom. And then our flagship summit is in October 18th and 19th in Boston. John Maraganori will be speaking. I think everybody in biotech knows John. Nubar Afeyan from Flagship, and author Megan O'Rourke, a best-selling author on disease and amazing poet too. So really hope to see some of you at some of those. Thanks to Foundation again for sponsoring this. Thanks to all of you for being there. Rick Burke will be happy that Angus gave you all that plug, but he's going to be even happier that each and every one of you showed up. So thank you so much, and have a glass of wine or a beer on us. Thanks so much. The sliders are good too.